Yes, if you've just turned in the channel by accident, no, this is not the Sochi Olympics. You're watching the Boards and Bio Briefings right here on ThinkTech Hawaii Television. And today, we're privileged to have as our guest Dr. Richard Allsopp, who is a professor at the J.A. Burns School of Medicine. I always have to remember JAP-SUM. Very good. And we're very privileged to have Dr. Allsopp with us because he's going to talk about something that may be of interest to you. Longevity. Yes, longevity is a hot topic. Who doesn't want to live longer? And we're going to be talking about his research in a subject called telomeres. No, they're not little crawly things that go around behind the closet. Telomeres are right now well and alive inside you, and we're going to find out what effect they will have on your body, what effect they will have on society, illness, diagnosis, treatment, and prognosis, and fortune telling, and various other things that are much of interest to almost everybody who's watching this show and others who are not watching, and we hope you'll tell them right here. Remember, in Boards and Bio Briefings, you're hot, we are not. Our guests are definitely hot, and let me introduce Dr. Richard Allsopp, and welcome.
Pleasure to be here.
And let me start off by asking, first of all, a question. What is aging, and how does it work in general?
Well, aging is the gradual deterioration of our bodily functions at all levels, at the organ level, at the cell level, at the molecule level. And this continues on gradually with age and ultimately increases the risk for death as we age, and is what limits the lifespan of humans as well as most other species on the planet.
Now, one of the things you hear in common, especially on non-technical or non-biological radio or television, is free radicals. Now, I always thought that with the demise of the Soviet Empire, the radical parties are gone. What is exactly free radicals? Are they guys running with guns or what?
Well, in our bodies, they're like little mini guns, little molecular guns that can actually damage other molecules, bigger molecules like DNA, important encoding molecules like that in the body, and proteins in the body, and stop them from functioning properly. They can, if there's enough of them, they can even kill the cells in your body.
So what exactly do we mean when we talk about oxidative damage? What does that mean?
So what it means is those free radicals, which are generated from oxygen, which and one of the main sources of that is the mitochondria, the powerhouses of the cell. They are getting out, and they are physically reacting with proteins and DNA in the body, and chemically bonding and altering the structure, and through that the function of proteins and DNA and other molecules in the cell.
So when we talk now in the popular press, when you hear on late night talk shows, usually about 2 o'clock in the morning, we've got just the lotion that's in motion with a potion for curing free radicals roaming around your body. What exactly are these additives or food products or pills or natural herbs? What are they exactly that people are offering sometimes?
Yeah, some of the things are, some of these molecules in components are vitamins like vitamin E and vitamin C that have been around for a long time, and there's newer and better ones coming out on the market all the time as well.
Well, is this basically the theory behind Linus Pauling's ideas of vitamin C maybe 40, 50 years ago when he got the Nobel Prize? You know, he advocated the heavy use of vitamin C as almost a cure-all for everything.
I don't know if at the time they thought it was as good an antioxidant as it turns out that it can be. Vitamin E, I think, is even better, but yeah, there's a lot of free radical antioxidants, and there's these enzymes in your body like superoxide dismutase is one that the body actually makes that can actually quench and soak up these free radicals as they're produced.
So what you're introducing is basically the idea that your body is probably the best recreation machine for healing the body.
It certainly does a pretty good job of that, yes.
So let me segue right into your topic. What are telomeres and what do they do?
So telomeres are genetic elements that are at the ends of all chromosomes in a human cell. So the human genome is broken up into 92 chromosomes, and these are linear pieces of DNA. And what the telomere is, it's the very tip of each chromosome that acts as a cap.
Let me perhaps make that a little clearer to our audience with picture number 1 there. There you go. So there are the telomeres, I'm sorry.
Yeah, so the telomeres are what are lighting up sort of at the feet and the head of those chromosomes there. And they, with age, gradually get shorter and shorter, in particular in cells that divide a lot, like blood cells and skin cells. So one of the problems that we're faced with is, as humans, is how to counter that process, because what happens is if the telomere gets too short, then it can't function as a protective cap, and this can cause instability in the genome, and that can cause damage to chromosomes, and ultimately it can cause a cell to senesce.
Segues right into my next question. What is cellular senescence?
Cell senescence, it's also known as the Hayflick limit, after Len Hayflick, who discovered the process in human fibroblasts about 40 years ago, which defines the finite, limited capacity of normal human cells to divide. So a normal human cell, a fibroblast or a skin cell or blood cell, what have you, most types of human cells can only divide a limited number of times in a tissue culture dish, like say 80 or 90 times. And what was discovered about 35 years after Len Hayflick's seminal discovery in the 60s is that telomeres gradually shorten as these cells divide in culture. And ultimately what happens is the telomere there's so much DNA lost from the telomere, it can't function properly as a cap. And that signals the whole senescence pathway. So what senescence refers to is the terminal inability of the cell to divide. It can no longer replicate itself. So it's not dead, it's not necessarily dead, but it can't make more cells. It can't renew and replenish organs, tissues of the body anymore.
So is there a natural process or natural additive that used to be perhaps taken previously before we understood how the whole process works? Was there a natural additive? Was there a herb, a Chinese medicine before that?
No.
That people really believed in?
To stop senescence?
Yeah.
No, I don't think so. I mean, if there was, I'd be taking it.
So that's why all the big money is going into this research. Mm-hmm. Well, I mean, not for you, of course, but I'm talking about in general. In general. In big pharma is really hot on the trail here.
It sure is. Sure is, yeah.
Well, let me ask you, what characteristics do cells exhibit when telomeres get shorter? I mean, just before the cells reach senescence.
Before the cells hit senescence, well, you know, they really don't. They do change a little bit in their function and things as they approach senescence, but it's really that transition to senescence where you get profound changes in how the cell can function. It becomes, one of the things it does is it secretes cytokines from the cell that sort of cause inflammation. And we talked about free radicals, so that can actually cause generation of free radicals, things that you don't want too much of within a local microenvironment. So one senescent cell is not good, but it actually can have additive effects by secreting these cytokines to all the cells that are in the vicinity, the near vicinity of that senescent cell.
So one of the biggest boons actually to plastic surgeons is perhaps the decrease in the synthesis of procollagen for ladies among our watching audience. And immediately noticeable. How would we notice that on someone's face?
Well, you know, it can cause wrinkles.
The W word. The W word. Okay. So actually, the skin is stretching and looks older, goes into folds.
Yeah. Well, we talked about cross-linking of proteins and things that free radicals can generate, and that ultimately can manifest as wrinkles. And, you know, It's something that has to be constantly battled.
So the
as,
protection
as
for
older we
the
get.
cells is gone, and they become then exposed, I guess, to the dehydration effects of the sun and everything else.
Well, there's different causes of the wrinkles. It's not all due to telomere shortening. I think the older you get, I think the more involved telomere and senescent cells can play. But there's immediate damage from sun. If you get exposed to the sun too much, even at a young age, you might not have a lot of senescent cells, but that can also cause wrinkles in the skin as well. So different causes for the same effect.
So if you can actually stop the shortening of the telomeres, will this prevent replicative aging?
It will prevent replicative aging. That we've shown to be true in tissue culture, and actually in mice as well. So in the tissue culture, you can reactivate an enzyme called telomerase, which functions to stop telomeres from shortening. It replicates telomeres as the cells divide. So it's an very important enzyme to have in order to maintain a cell's replicative potential. So stem cells, for example, in the body, which replenish organs daily, as normal bodily processes go on and cells turn over, these stem cells often have telomerase in them, whereas other more differentiated cells, like a skin cell, might have no detectable telomerase.
Now, allow me just to digress for a second. You mentioned mice. Now, I noticed that sometimes some of the proteins get expressed or get used different animals for producing new products, new bacteria, new viruses, new molecules, and so on. Why mice?
Well, mice are a great organism to study for aging for a number of reasons. I mean, one, they're really small. You can fit a lot in a cage in a laboratory environment. So you can have sufficient numbers of these animals to do statistical analysis. The other thing is that, or even more important, is the fact that they have a relatively short lifespan. So they only live about 2 years or so. And they reproduce relatively quickly to generate more mice for other experiments. So reproduce fast, don't have a long lifespan, so you can study the effects of whatever you want to study on the effect on the lifespan.
So instead of getting a human expression of the same product, waiting around 70, 80 years for someone to age, you can get something pretty quickly.
You're much quicker.
But what about the differences in species?
Exactly. So there are limits there as well. I mean, the mice are good. They're mammals like humans are. So they might be something a little bit better than, say, yeast or like a fruit fly, but they're still not humans. So they have a different genetic genome, slightly different than humans. They have a different number of chromosomes, but nonetheless, I think there is some relevance there to human biology. But whatever you find in mice, that's the first step to indicate that it might have an effect in humans. Then, of course, you need to do the clinical trial in humans to make sure that it does.
Well, let's hold that thought right there. We're going to take a break to our sponsors. You're watching Boritzer's Bio Briefings right here on ThinkTech Hawaii. Our guest today, Professor Richard Allsopp at the JAB School of Medicine at the University of Hawaii.
Castle and Cooke, Hawaii. Investing in Hawaii, creating communities, and providing for the needs of our state. Collateral Analytics, empowering the real estate industry to make better informed property investment decisions. The Foreign Trade Zone, bringing the benefits of the Foreign Trade Zone programs to Hawaii businesses and entrepreneurs. Galen Ho, a senior executive of BAE Systems, a global defense, security, and aerospace company. Hawaiian Electric Company, and its affiliates Maui Electric and Hawaii Electric Light Company on Hawaii Island. The Hawaii Energy Policy Forum, incorporating diverse perspectives to design a flexible and forward-looking energy strategy. Hawaii Energy, the state's energy and efficiency program created to help Hawaii's residents and businesses adopt a clean energy lifestyle. Hawaii Gas, helping Hawaii achieve its transition to clean energy and a better energy future. Hawaii Pacific Health, bringing technology and teamwork together to transform healthcare in Hawaii. The High Tech Development Corporation, attached to DBED, is the state's leading technology agency. The SHIDLER Family Foundation, supporting educational, cultural, and charitable organizations, including ThinkTech.
We're watching you. We hope you didn't go to the refrigerator and pick up some telomerase while we were on a break. Our guest today is Professor Richard Allsopp, whose research in telomeres well known, and we're discussing the effects of the telomeres on research into aging and treatment of a variety of diseases. And tell me, what is exactly telomerase?
Telomerase. So you have the telomere, which we talked about last time, which is the degenerate cap that caps the ends. Telomerase is a protein. It's an enzymatic complex, an enzyme that recognizes the telomere, and it has the function to add more telomeric DNA onto the end of the telomere. So it can actually stop the shortening of telomeres, which would otherwise occur as the cells divide, because there's a problem that the normal DNA replication machinery has in replicating the end of the telomere. It just can't quite make it all the way to the end. So there's little bits that are left unreplicated in any cell that divides with no telomerase. If telomerase is present, because it can extend the end of the telomeric DNA, then it overcomes that problem.
Well, can telomerase use as a product to extend the cell lifespan?
Something that could transiently activate telomerase could do.
Well, can you possibly speculate on other future biomedical applications of this new technology in resetting the telomeres clock?
Oh yeah. Well, there's been a number of diseases that have been associated with mutations in telomerase. That some are sort of related to aging, some surprisingly not so. For example, lung fibrosis has been shown to be associated with certain mutations in one of the essential components of telomerase, called telomerase reverse transcriptase, which is the catalytic component, which is a key essential component to the function of telomerase. So if you have mutations in the gene that encodes this telomerase reverse transcriptase, which I will refer to as TERT, acronym called TERT, T-E-R-T, then you are then predisposed to get lung fibrosis. So something which could reactivate telomerase, or the good copy of telomerase that someone might have, it could compensate and prevent, theoretically, the deterioration of the lung that's associated with pulmonary fibrosis. So that's one. And then another one is heart disease. Heart disease, which is an age-related process, has been shown to be associated fairly strongly with reduced telomere lengths in the blood.
So is it really possible to revert old cells into young cells and once again?
Yeah. It sure is. I mean, we've done it in tissue culture. You can reactivate telomerase in an old skin cell, and it will not senesce. It will stay youthful. It'll keep dividing and dividing.
No, but the question comes, you can look like a 20-year-old, but can you think like a 20-year-old? Or do you want to, really?
Can you think like a 20-year-old? Well,
that may require a different expression of the bacteria or the molecule.
It might. I don't know if you necessarily would want to think like a 20-year-old, but
Okay, so if some aspect of human aging is directly linked to short telomeres, does the gradual shortening of the telomeres coincide with the long-term aging process over a lifetime?
It does, and it's been associated, particularly in the tissues and organs in the body that divide a lot, like the intestine, the skin, the blood. And in the blood, for example, if you take blood samples from people that are 100 years old, their telomeres are so short in the blood that they are approaching what you find in senescent cells, like a pure homogeneous population of senescent cells. These blood cells from centenarians are verging on the border of becoming senescent.
Oh, I see. So do you see a scenario where telomeres can be the Achilles heel of cancer?
Well, they certainly can maintain. If telomerase is key in cancer and the immortalization of cancer cells. So cancer cells are typically, not always, but typically they are immortal. They've overcome the senescence checkpoint. They don't senesce because their telomeres don't get too short that they can stop functioning. And the reason that they don't senesce is because telomerase has somehow, in about 90% of cancers, been reactivated due to a set of mutations, most of which have yet to be discovered. But it's nonetheless, it's a rare event. But if it happens, then that endows the cancer cell with immortalization.
Oh, I see. So what you're saying is that if you inhibit the telomerase, then you can restore the senescence program in the cells?
No, once the cell hits senescence, it can't be reversed. But if you reactivate telomerase before it's senescent, then it won't get senescent. It'll just keep dividing.
So you're saying that it's a little more complex to say that we can find a therapy cure for cancer in the telomerase?
Oh no, I think that if you could inhibit telomerase in a cancer cell, that it theoretically could cause the cancer cell to senesce. The interesting thing about cancer cells is that most of them have very short telomeres. So if you just inhibit telomerase for a little while, it'll cause a lot of those cancer cells to senesce. So it's in theory, it's a very logical and sensible way to potentially treat cancer.
I see. How specific a targeting would you have to be to attack cells like the reproductive cells?
So yeah, those are cells that you don't want to target like in a cancer therapy, but we believe that they might be able to bounce back relatively quickly because they have long, in addition to having a lot of telomerase, they also have long telomeres. So they can withstand certainly a short duration of time with no telomerase. So.
Oh, I see.
Yeah.
So it's almost like giving them a lead coat when they're getting exposed to radiation.
That's right.
For certain cells.
Yeah. Mm-hmm.
So could there be another substance that could be used as a screen when applying this therapy? Would it have to be a two-part or a two-step or two-stage kind of treatment if you're trying to imagine what kind of treatments we could invent for cancer using the telomerase?
Well, I mean, I think that it certainly would supplement other current therapies for cancer. I think a multi-pronged approach where you have a telomerase inhibitor type of compound with, for example, another compound that stops the spread or the metastasis of a cancer, and then maybe even something else that can prevent the growth of blood vessels in a cancer. So, just as an example, these different types of combination of treatments, I think, ultimately will work best.
To your knowledge, is anybody doing actually clinical trials with telomerase?
Well, there's one company in California called Geron, which has got a couple clinical trials ongoing for a telomerase inhibitor compound that they developed, and so they're working hard at it.
So this is basically a totally different approach and has been used till now in cancer treatment in terms of what till now we've been using chemo or radiation therapy or radiotherapy. Mm-hmm. So this is a totally different approach to the issue.
This is a totally different approach, a very novel and different approach to the treatment of cancer from conventional ways.
And in terms of envisioning this kind of treatment, in terms of being the patient, mm-hmm. What kind of length of time of treatments and what about frequencies? Is anybody talking yet about that type of issue? In terms of the intensity, the length of time, because today we tend to talk about tumors and cancers in terms of how advanced, how progressive the cancer is, how far spread it is it metastasized or not. When you're looking at telomeres, do you view it from the same type of stencil or viewing
Yeah.
microscope?
Well, a lot of factors play into the time that it would require to treat a cancer with, say, like a telomerase inhibitor, because it's not just about stopping the telomerase and the telomeres are going to start shorten. It's about how accessible the cells are to this drug, things like that, which could, have to be taken into consideration. But it could be, since most cancer cells are relatively have short telomeres, it could be as long as it takes for the cell to divide a couple times or maybe even one time with no telomerase, which, if in a rapidly growing cancer could be within a day or two.
It might sound, very strange. I'm going to ask you, what happens if you leave your telomere therapy lotion and potion in the closet? Is there a danger to someone in your household accidentally drinking it or
Your
taking the medication?
To the telomerase inhibitor?
Yeah.
Well, there's always risks with any therapy, but the side effects for this type of compound, just purely based on the biology of the effect of it, which is on telomeres and telomerase, is the cells in the body which have and require telomerase, which are the stem cells and the reproductive cells of the body. So, but as we talked about a little bit before, those cells happen to have, luckily, relatively long telomeres. So if you hit them with this drug, they'll be able to go some time before there's any deleterious effects, because they have such a big buffer of telomeres in them.
So it's actually this form of reprogramming of the cells is totally different than the approach that's used in chemistry or in pharmaceuticals that use chemical compounds instead of biologics.
Well, the reprogramming is where you add DNA to a cell and or a certain piece of DNA that encodes a gene that is then produced in the cell. Whereas, what we've been talking about with telomerase inhibitors, we're talking about small molecules. So yeah, those are quite two different.
Okay. Can you think of any other safety issues involved in telomere therapy?
Telomerase cancer therapy, I don't think. It's going to be, a lot of it's going to boil down to the nature of the small molecule as well, what kind of toxicological effects it's going to have. But that'll come out in the screening process and the toxicological tests.
Okay, we're going to take this opportunity, take a break and thank our sponsors and identify our station. You're watching Boritzer's Bio Briefings right here on ThinkTech Hawaii. And our guest today is Professor Richard Allsopp, specialist in telomere research, and giving us the reasons why we're going to live a lot longer, at least until tomorrow. At least stay with us till our end of a station break.
Hi, I'm David Day. I'm the host of ThinkTech Asia. What we do is produce shows that have to do with international business, foreign policy, national security, and geopolitics. We run our shows on Thursday afternoons at 4 o'clock. I'll see you.
Aloha, I'm Maria Kashem of ThinkTech Hawaii, and I want to tell you about our ThinkTech talk shows. If you didn't know it, ThinkTech streams video and audio for all of its shows live on the internet from 2 to 5 p.m. every weekday afternoon, and we replay them all night long on ustream.tv. Visit thinktechhawaii.com for our live stream and YouTube links. Raise your awareness on ThinkTech. I'm Maria Kashem, and I'll see you there.
Aloha, I'm Jay Fidel of ThinkTech. We have some news for you. In addition to our ThinkTech TV show and our Asia in Review show on Olelo 54, as of January 1st, we're adding Community Matters to play also 2 hours a week. Check out thinktechhawaii.com for the specific times of each of these shows. We hope you enjoy all three. Mahalo, I'm Jay Fidel.
Welcome back. We've been watching you. We hope you'll continue watching us right here on Boritzer's Bio Briefings. We're talking about the telomeres and the telomeres. I don't know why I keep saying telomeres, but it's telomeres and the life extension, longevity, cancer treatments, all the good things that this branch of science, molecular biology, that's developed over the last, how long would you say it's taken for it to come to fruition in terms of funding, in terms of interest, in terms of numbers of people involved?
Well, yeah, telomeres were discovered in Drosophila and maize corn about 60, 80 years ago. And so then, it took some time before the actual enzyme telomerase was discovered. That was discovered in the mid-'80s by Carol Greider and Liz Blackburn, who got the Nobel Prize for that discovery. So, it wasn't until that enzyme was discovered that the whole field started taking off. So it's really been since about the mid-'80s that it really started taking off.
I see. So actually, you consider that a fairly short time.
It's a relatively short
time.
time.
Science research.
Oh, yeah.
I know that people hear a news story or they listen to a show like ours today and the discussion about longevity and telomeres, and they think that tomorrow there'll be a drug, there'll be a potion, a motion, or a lotion that will take care of things, but it just doesn't work that quickly.
It doesn't, but it does and it doesn't. I mean, if you're talking about an FDA-regulated drug, there's a whole process and series of steps that have to be done, rigorously monitored process with clinical trials and all this, very expensive. $1 billion later, 20 years, you have a drug. But if you have an extract, which is like a natural product that people are already consuming that happens to have some, say, the ability to activate telomerase in cell and stop the cell senescence, potentially could be much quicker to the consumer.
Yeah, I want to interject something before we continue in the discussion of telomeres. One of the things that are done quite frequently outside the United States, where you don't have the same structure as the Food and Drug Administration in terms of approval of chemically based pharmaceuticals, is that you can, if you're a physician and you have an experimental treatment center or hospital or a cancer center, so you can try things that are not approved by the central agency, but things that are scientifically been proven to be effective, and with the consent of patients coming to your specific facility who understand that they're being part of a clinical trial, these things are tried out in situ and in vivo, actually in real life, as opposed to going through the process of 6 to 8 years of drug development, testing, and so on. Do you think that this, especially when it comes to molecules and when it comes to biologicals, do you think that this is a more realistic way of trying out things before they seem to work or don't work over a period of use? So we don't see the commercials on television. If you have suffered, death or chronic illness due to taking this particular drug. Yeah.
Well, I mean, as rigorous as the FDA process is, there's always things that they can miss. So, that's why it's so rigorous. They have 2 or 3 clinical trials that any pharmacological agent has to get through in order to get it on the market. But even then, there are things that can be missed. So I think you pretty much have to do, certainly for a drug which is got a lot of potent activity, that you pretty much have to do that.
Well, I think that when we are talking about the standard chemical pharmaceuticals, you're talking about proving to an agency, whether it's the FDA in the United States or any other control agency, that a high percentage of people will benefit from that drug. Given the caveat that if you have this A, B, C, or D, you shouldn't take it. If you shouldn't contraindicated drugs together with it, you shouldn't do it when you're asleep, you shouldn't do it when you're awake, you shouldn't do it when you're eating.
Right.
So all these caveats, because you're trying to get the highest percentage of effectiveness and lack of toxicity or contraindication of the drugs. But when you're using, and correct me if I'm wrong, but if you're using biologicals or protein-generated solutions, you can almost have personalized medicine coming out of this type of philosophy.
Possibly, yes. I think that there's a lot of promise there, because it's you're consuming biological, like something that's already commercially available, but and by combining some of these biologicals, you might have something that just happens to work really well in activating telomerase, preventing cell senescence. So yeah, I think there is a lot of potential there.
So it's almost, you know, it's rather ironic. We're almost going back to an era pre-pharmaceutical industry where grandma's lotions and potions seemed to work. She didn't know why they were working, but her mother and their grandmother told her that these worked for particular ailments. And it didn't matter if they didn't work on the whole village. If they worked on your child or your parent or your uncle, that was enough proof that of what they needed. It wasn't really important if you had the consensus, but maybe the historical in your family, the historical proof that something worked perhaps was indicative of its potency and effectiveness. So, let me go back if you don't mind. So, if it's possible, or is it possible that telomerase can lead to drug resistance?
Telomerase lead to drug resistance. Not itself. Are you talking about the enzyme, the inhibitor?
Yeah.
Could you get resistance to it? Yeah, you could get resistance to it. There's a small percentage of tumors that actually can replicate telomeres without telomerase. So it's possible that through chronic exposure of a patient to the telomerase inhibitor, the cancer may initially regress, but maybe some of those cells might activate this alternative pathway, as it's called, this alt mechanism of replicating telomeres. So that's always a worry, it's always a concern, but I mean the question is how long and how is it going to take years and years, decades, or is it going to take months for this to resistant mechanism to show up, and is it dependent on the type of cancer? So there's a lot of questions that we just don't know the answer to.
So actually the tumors might become even more potent as a result. Possibly in some people.
Well, these telomerase negative tumors, interestingly, they can't divide quite as well as the telomerase positive.
Really?
Because the alternative mechanism is not as efficient as telomerase itself. So the jury's still out on how these resistant cells, even the jury's still out on even if they will arise. It hasn't been put to the test yet.
Hold that thought. We're going to be right back. You're watching Boritzer's Bio Briefings right here at ThinkTech Hawaii. My special guest today, Professor Richard Allsopp, specialist expert in telomeres.
I'm Hong Jiang, host for Asia in Review on Tuesdays.
And I'm David Day, host for Asian Review on Thursdays. Both of us broadcast our respective shows at 4 p.m.
And my topics tend to deal with questions related to environment, culture, history, and sometimes human rights.
And my shows tend to be on international business, foreign policy, geopolitics, and national security. And you can watch our shows live on the ThinkTech website at thinktechhawaii.com.
And you can also watch us on YouTube or Alalo.
So come join us on Thursdays at 4 p.m. I'm David Day.
And on Tuesdays at 4 p.m., I'm Hong Jiang.
Aloha.
Aloha.
We're back. We're watching you, and we hope you're watching us as well. Our special guest, Dr. Allsopp, is here to discuss telomeres. And let me get right back to the subject. Since some cancers don't express telomeres, how are they maintaining their telomeres since they don't express telomerase?
There's this alternative pathway, so ALT. So what it does is it actually is a mechanism where a telomere can sort of recombine with another telomere and sort of act as a template for the normal DNA replication machinery and other recombination proteins to extend a telomere. It's a much less efficient process, and it doesn't occur all the time. It sort of occurs when a telomere gets really short. It might get repaired. Sometimes the cell may not repair, and you might end up getting a lot of a senescent cell, whereas telomerase positive cells populations are generally, the number of senescent cells in a telomerase positive cancer, say, are relatively small, but in an alt mechanism, you do get the odd cell that gets these critically short telomeres because it's not super efficient, but it's a DNA recombination pathway.
Have any telomerase agents been identified, therapeutic agents?
Telomerase inhibitor.
Yeah.
Well, as I mentioned, Geron Corporation in Menlo Park, California, have a proprietary molecule that they've discovered and published on. It's actually associates with the RNA component of telomerase. So that's another essential component of this telomerase protein. So RNA, the telomerase RNA, is actually what serves as a template to extend the end of the telomere. And this molecule that Geron Corporation has developed will block that RNA component from functioning properly.
What do you think is the next step in the research towards the treatment of cancer with the whole telomere community?
Well, I think that we're only just beginning now to sort of identify what some of these telomerase inhibitor compounds are. So I think that as time goes on, I think people will naturally find better and better inhibitors that can affect tumor cells. But it's going to be a gradual process. It took Geron Corporation over 20 years to develop their inhibitor. So, new technology is always changing, always developing. The next one could be around the corner.
So you think that detection of telomerase activity will be useful in cancer diagnosis?
Yeah, it could be. It could be, as could telomere length. The length of telomeres. If you have cells which are, as I said, tumor cells have relatively short telomeres. If you have cells in your body which are abnormally short, it could be an indication that telomerase is active. But the enzyme also could be tested for. There's an assay where you can actually measure, you can make an extract from the cell and mix it with a substrate of telomerase, and it'll fluoresce. If it's extended, you can amplify it, and then if it amplifies, it'll fluoresce, and then you can detect it. And so that's one method to detect telomerase in cells, and it could be theoretically could be used to screen for higher levels of telomerase or spikes in telomerase that could be associated with possibly. I think this would have to be done in conjunction with other tests that are out there for cancer diagnosis.
So how long do you think it will be before telomerase can have a significant medical impact, such as cancer screening or telomerase therapy?
Yeah, I think cancer screening might come a little bit sooner, maybe 5 years. Cancer treatment could be maybe anywhere in the range of 5 to 10 years. It's hard to say, though. It's hard to put specific numbers on these kinds of predictions, but that's sort of some optimistic timeframes.
Well, there's I'm sure that you haven't been living on Mars in the last year, and so you know about the whole brouhaha regarding the doping in the Lance Armstrong case and the whole bicycling team, and in effect the whole sport of professional bicycling. I'm not so curious about finding out if doping is finding out. I'm actually curious to see if all that doping is actually part of the process. Maybe it has an effect on the telomeres themselves, rejuvenating the ability of the athlete to perform, so to speak, at the superior levels.
Yeah, well, who knows? It could, like growth hormone, these sorts of things. I honestly don't know if it does or if it doesn't. I'm not sure if it's been tested. But there's no...
I'm thinking more in terms of diagnosing the potential of an athlete. We have all these, America is very crazy about sports, professional football, basketball. I think there was just now some kind of a basketball championship. Everybody was betting money, $1 billion against, to see if they could come up with the final winners of the basketball team. So do you think that telomerase has the potential for identifying the super athletes of the future?
I think it definitely has the potential. I mean, I think one of the things that hasn't been looked at and would be really exciting to look at is genetic variants of the telomerase gene, which may lead to, for example, a more active telomerase or a more stable telomerase kind of scenario, which very well may be out there, but waiting to be discovered. And if there's people that have these protective sort of anti-aging, pro-longevity effects of telomerase, then it could have some indication that this person over here who has it may have a little bit advantage.
So, just as a final question, what do we need to do? How to move faster pace forward so we can address many of the unanswered questions, for instance, in aging research?
Well, I think that what we just talked about actually is very interesting. The discovery of variants of telomerase that may be more potent. I think the development of telomerase activators to activate telomerase in a safe setting through these biologicals, medicines that we were talking about. I think and cosmeceuticals and things and nutraceuticals hold a lot of potential. So I think, but also all of this takes money. So money, the more and interest, people, manpower. I think you can really accelerate the process.
I want to thank our special guest today, Professor Richard Allsopp from the John A. Burns School of Medicine at the University of Hawaii. And we've had a really mind-blowing, revealing discussion on telomeres and telomerase. And I want to thank also Professor Allsopp for being here. And I also would like to thank the staff right here at ThinkTech Hawaii that week after week, day after day, puts on the programs that you're watching. Special thanks go out to our producer, Jay Fidel, who's put the whole shebang together. You've been watching Boritzer's Bio Briefings right here on ThinkTech Hawaii. Until next week, remember, you're hot, we're not.